My name is Matt Tomlinson, and I'm a field application scientist for Sartorius Bioanalytics. And I'm going to talk to you about technologies and solutions for the development of novel biopharmaceuticals from molecule identification to cell line development. And I'm going to start with a statement, and that is that more hits doesn't necessarily mean better therapeutics, but better characterized hits can lead to better therapeutics. However, to achieve this, you require tools that allow for both the acquisition and analysis of large data sets. And I'm going to talk about this in the context of immune cell mediated killing. Now, this encompasses a variety of approaches, such as adoptive T cell therapy, chimeric antigen receptors, tumor infiltrating lymphocytes, NK cells, and soluble T cell engagers. Now, if we look at an example immune cell killing assay, in this example, we have NK cells. There are a variety of different reporters in which we're interested. So we have our targets and our effector cells, and we're interested in immunophenotyping those. We're interested in functional markers, such as markers of activation in NK cells, CD69 and or CD25. We're interested in cell death, of course, so we're potentially going to be looking at cell membrane integrity dyes and markers of apoptosis. Cell count is, of course, important as well. And we're also potentially interested in cytokine profiling. So looking here at pro-inflammatory cytokines, such as interferon gamma, and pro-apoptotic proteases, such as granzyme B. Now, one approach to assess immune cell killing is live cell imaging and analysis. In this example, we have an adherent target culture. We're running a 3-color analysis. So here we have A549 target cells that have been transduced to express a near-infrared nuclear restricted reporter. In this assay, this is pseudo-colored blue for visualization. These cells have been cultured in the presence of anti-CD45 labeled PBMCs, and the anti-CD45 antibody has itself been labeled with a Fabflor 488 reporter. And these cells have been cultured in the presence of a Nexin-5 orange to assess apoptosis. Now, here we have non-activated PBMCs cultured with the target cells, and what we see is proliferation of those targets. We see increasing cell density and not much in the way of killing. However, when we look at activated PBMCs, we see something quite different. We don't see proliferation of the targets, but we do see proliferation of the CD45 positive PBMCs. And we also see an increase in a Nexin-5 orange signal. We can then plot this information on a population level, and we can see that the target cells proliferate when they're cultured with non-activated PBMCs. However, when they're cultured with increasing numbers of activated PBMCs, we see a decrease in proliferation. And conversely, at the highest PBMC density, we see the highest amount of proliferation. When we look at apoptosis, we can see little killing when the PBMCs are non-activated. However, with an increasing effector to target ratio, we see increasing apoptosis, and we can also get information about the timing of this event. So we can see that killing starts around 48 hours into this assay. We can perform a cell-by-cell analysis on the effector cells, and we can look at the percentage of cells which express CD45. When they're activated, there is a consistent expression level. However, when the PBMCs are non-activated, we actually see CD45 dropping over time. We can also assess immune cell killing utilizing live cell imaging and analysis for suspension target cells. And here we can also utilize cell-by-cell analysis on both the targets and the effectors. In this assay, we have Ramos target cells. These are being transduced to express a red nuclear reporter, and they're cultured with PBMCs in the presence of a Nexin-5 green. When the PBMCs are non-activated, we see proliferation of the target cells and not much in the way of cell killing. However, when the PBMCs are activated, We see a strong proliferative response from the PBMCs. We see swarming of the target cells, and we see an increase in an Nectin-5 signal. We also see a morphological change in the effector cells as well. We can see them go from this rounded morphology to a more elongated and larger cell shape. From these images, we can perform single cell segmentation, and then we can get additional information on a per cell level in a kinetic manner. And we can plot that at each individual time point, as you can see here. So we have here our target population. These are our Ramos red positive cells. And here we have our red negative effector cells. We can also assess apoptosis here in the different populations. And population level analysis allows us to look at target cell proliferation. We see at the highest effector to target ratio, we have the least proliferation of the Ramos cells, but we have the highest target cell death. We can perform an apoptotic index analysis here based on the single cell segmentation. We can also assess immune cell proliferation in this assay, and we can see that the highest effector to target ratio, we have the highest proliferative response. Now, this temporal analysis gives much greater insight into the assay and the timing of events. And this therefore allows us to better characterize the treatments, and we can make more informed decisions upon which treatment is going to give the most effective killing response. And we can obtain significantly better information in terms of the timing of that response. What we can then do is transfer this assay to more multiparametric forms of analysis, such as flow cytometry, to obtain further insight into the mechanism of action. And in this example here, we're looking at an antibody-mediated cell killing assay, and that is, of course, entirely amenable to a live cell imaging approach as well. Here, we're looking at CD20 Ramos positive cells versus CD20 negative HL60 cells. We have our PBMCs, and from here we can assess the dead cells versus the live cells, and we can also look at dose response curves as well. Now I'm going to walk through how we would utilize the IQ3 to analyze a potentially immune cell killing data set. In this example, we're actually looking at immune cell activation. So we'd set up our plate here. We could choose our channels of interest, choose our wells we want to sample. We can then determine which data corresponds to which well. Can then gate on our beads and our cells. We can perform singlet gating. We can also look at the beads. We can look at our live cells, our dead cells, and then we can perform immunophenotyping. We can look at our CD3 positive population, and from that we can then look at the CD8 and CD4 positive cells. We can then look at markers of activation such as CD69, and we can then perform heat mapping, so we can see which wells have the highest proportion of activated cells. We can then look at a profile map, and this allows us to perform hit finding. So what we can do is adjust the parameters and the criteria which we consider to be a positive hit using these slider bars, or we can manually input those values, and we can then hone in on the different wells that contain our hits of interest. Now, this was one single plate. When we're looking at multiple plates, we can utilize the panorama features of the Foresight software. So we can look at this same analysis. We have the same gating strategy, the same heat maps, the same profile map. And we can now start to look at different plates in our experimental data set. And we can see how those parameters, how those criteria fit this particular plate. However, we also want to look at this in a comparative view, and for that we can use Panorama. So we can load in the plates for our assay. In this example there are 2 plates, but we can have many more plates in an assay, and we can adjust those parameters in the profile map, and we can do this now for our entire experiment rather than a per plate basis. And this allows for very quick and easy hit finding and better characterization of those wells. So I've shown you how to perform that hit finding using the Panorama software. And here we're looking at a heat map that's been generated in Panorama. And this is an ADCC assay again. And we're looking at CD20 positive and negative cell lines. And here we're actually looking at different antibody engineering approaches. So we've used here rituximab, and we've got our IgG1, IgG2, and then engineered variants of the IgG1. We have a fucosylation deficient variant and a glycosylation deficient variant. And what we're looking at is cell killing of these different plates, these different treatments. We can see that the IgG2 and the glycosylation deficient variants don't induce much in the way of cell killing. However, when we look at the fucosylation deficient variant and the IgG1, we do see a significant drop-off in cell viability. So what this really allows us to perform is a mechanistic analysis, and we can then characterize our antibodies better to be able to then induce more potent cell killing responses. And then to finish up in the functional studies, we then want to take this to something that's more physiologically relevant. So we can utilize a tumor spheroid immune cell killing assay. Here we have A549 cells. These have been cultured with PBMCs. The A549s express a red nuclear restricted marker. We can see when we look at non-activated PBMCs, we can see a proliferative response from that. In that spheroid, we see an increase in red fluorescence. However, when the cells are cultured with activated PBMCs, we see something quite different. We see that red fluorescence signal start to drop off and diminish over time as those cells are being killed by the activated PBMCs. And we can then plot this information, and we can show this decrease in red fluorescence intensity following addition of activated PBMCs. Now, we've spoken a lot about functional studies and how we can determine the action of different treatments on inducing cell killing. I'm now going to change track slightly and talk about cell line development and using high throughput analysis approaches for this. Now, the first thing we want to do when we're developing a cell line is determine that the cell line is indeed a clonally derived population. And live cell imaging and analysis is ideally suited to this. In this approach, we would seed a single cell to a well. We could then identify colony formation, and then we can wind the clock back to determine clonality. And if we see in this movie here, these are MDA B231 cells, we can see a colony that's formed, and we can then run the clock back in reverse. We see those cells start to go down until finally we go from 2 cells and go to 1. So we can see that this is indeed a clonally derived population. Now, once we have our clones, of course, we then want to perform clone selection. So we can tighter the IgG concentration in each well, and we can then rank our clones based on IgG production. However, if you then start to look at cell density in yellow, and then the calculated IgG per cell, which is in red, you can see now that that ranking would indeed change based upon that additional analysis. And finally, we can then bring in a reporter of cell viability, shown in grey, and we can actually see that clones 6, 4, and 9 have much higher viability. And these now become much more interesting because they not only secrete a high amount of IgG, they grow well, and they are also viable. And then we can see that clone ranking dramatically changes when these other attributes are taken into account. And this was a screen of over 4,000 different clones. So it's a very high throughput assay and allows us to really focus in on those clones of significant interest. And with that, I'd like to hand over to my colleague, Dr. Helga Schnee.
Hi, also a warm welcome from my side. I'm Helge Schneer, Senior Marketing Manager at Sartorius. In the remaining minutes of this talk, we will discover BLI, and I have picked one bioprocessing application which should stand representatively for potential applications of this technology. Biolayer interferometry, or short BLI, is a label-free technology for measuring biomolecular interactions. Our instruments will monitor change in wavelength shift over time. This real-time binding measurement can be used to calculate on and off rates by plotting binding rates against concentration. Listed here are applications relevant in process development, monitoring, and QC. For instance, rapid quantitation of biomolecules in complex samples, potency assays of your therapeutic candidate, or rapid contamination detection in process samples such as host cell protein or protein A residues, or glycan profiling. Therefore, compliance with regulated environments makes it mandatory to support our users with a comprehensive GXP package as well. It includes even a biosensor lot surface to keep the coefficient of variation between sensor batches to a minimum. The chosen application is the relative screening of the glycosylation profile of biologics. Glycosylation is considered among the most important post-translational modifications when developing new biologics. Having a significant impact on product performance and variability, glycosylation is a critical quality attribute influencing product safety and efficacy. Protein glycosylation can affect isolation and purification steps, pharmacokinetic properties, and in vitro stability. The developed assay is based on pre-immobilized lectin on our microfluidic-free biosensor tip. It is dipped directly into the sample plate to bind proteins with the correct glycosylation. In a second step, the protein of interest is differentiated from the host cell proteins, and this signal can be even further enhanced to increase the sensitivity. The measurement is non-destructive, and the sample plate can be stored or used for further characterizations afterwards. In terms of throughput, up to 96 samples can be measured in parallel. The assay is easy to use and tolerates crude samples. This means that this allows to screen very cost-effective in early stages of development, where the numbers of samples might be a couple of hundreds or even thousands, rather than waiting for a later stage where only a few samples have to be characterized, but the likelihood of having a protein without correct glycosylation is higher. So identify the high-quality candidates earlier to prevent late failures downstream. Furthermore, we can combine a screening of the glycan profile with titer determination. And this allows us actually to identify not only the clones which produce the biologics with the right glycan profile, we find in the same assay the high producers, and this means we can now forward in the downstream development process the optimal candidates for the production of our biologic. You can also run titer determination in parallel with affinity ranking experiments. Through the off-rate measurements, antibodies with the optimal binding profile can be identified very easily. And here, this is BLI Technologies' strong value proposition, as we have a significant reduction of time in comparison to conventional technologies like analytical HPLC or ELISA by factor 25. And last but not least, you are not limited to the shown example. With our plate-based instrument platforms, you can measure, for instance, binding kinetics, which would be important for protein-protein interactions in general, or antibody-antigen characterizations in particular. But it would include as well off-rate rankings, or in the field of drug discovery when you measure small molecules against receptor binding. You can determine steady state affinity. This is very much important when you have small molecules which show a very fast on and off rate. So the equilibrium titrations might be the way to go. We have the sample quantification as seen. So to quantify the active target concentration of your analyte, which would include antibody quantification, clone selection, residues analysis, glycan profiling, or you simply use it as well for the development of your processes or the monitoring of processes by using concentration as your parameter. And you can do epitope binning. If you have large pools of IgGs for characterization and you want to find out if they inhibit each other or show cross reactivity or competition, you can run with a platform all three classical assays, the in tandem, the sandwich, or the premix assay. And with that, back to Matt.
So I'd just like to wrap up by going through the three different technologies which have been used to generate the data shown in this presentation. We have the IncuCyte SX5 live cell imaging and analysis system. This is an incubator-based system which is able to obtain images of cells on a repeating basis and then convert those images into data points. This allows for the kinetic analysis of dynamic processes such as immune cell killing assays in an optimized environment for cell growth. The SX5 is capable of obtaining up to 5 different fluorescent channels, with up to 3 channels being obtained simultaneously. The system can support up to 6 microplates in parallel, allowing for high throughput analysis in a live cell imaging context. Next, we have the IQ3, which is a flow cytometry-based approach for high throughput screening in a rapid manner. The IQ3 can process a 96-well plate in under 5 minutes and a 384-well plate in under 15 minutes. It utilizes a low sample volume for precious samples and has software, which I believe we've shown in the presentation, that is designed for hit discovery as well as better hit characterization. Finally, we have the Octet HTX. This allows the analysis of biomolecular interactions in a label-free manner at high speed and high throughput. It utilizes a fluidics-free dip and read technology, which allows for ease of use and high flexibility, and is able to quantify up to 96 samples in 2 minutes. So I think what we've shown in this presentation is indeed that better characterized hits are more likely to mean better therapeutics, not just more hits. And I'd like to thank you for your attention.